Microbial and Bioprocess Technology, video lecture 5. And this is very important, the basic one. Major types of microorganisms used in the fermentation process. This is the outline of the microorganisms and living cells used in the bioprocess. So we are going to have all the microorganisms, including the animal cell and the plant cell. Bacteria, for example, here. So this one is E. coli, starting from E. coli to fungi, plant cells, including algae. So because algae is one of the very important one for the production of oil, that is biodiesel production and biohydrogen production also. And hairy root plant cell cultures used for the secondary metabolite production, and animal cells for the production of monoclonal antibodies, and the growth hormones, peptides, and all the pharmaceutically important the components, and the human stem cells, artificial cells. And so these are the different categories of the different products from different organisms. The first table number 1 is typical recombinant E. coli derived therapeutic proteins. So all the components of the table 1 derived from E. coli, that is recombinant E. coli, starting from human interferon alpha, human interferon gamma, insulin-like growth factor, and the human granulocyte colony stimulating factor, growth hormone, human proinsulin, herodin, cellulose binding protein, all the very important components produced from E. coli strain. And so this is by using the different fermentation conditions. This is a 5 liter fermenter fed batch, and fermenter 2 liter volume fed batch, and fermenter 2 liter volume fed batch. So what is the different type of fermentation condition, and what is the name of the strain involved, and what are the products we are going to get it from the strains. So similarly, the table number 2, the recombinant proteins that commercially produced by yeast species, that is Saccharomyces cerevisiae. These are all the producers, that is Aventis, Novartis, all the biotech companies and pharma companies involved in the production of recombinant strains of yeast. The recombinant herodin and recombinant human insulin analogs and the recombinant human glucagon. So these are the very famous products, and these are all the producers, very famous producers related to biotech industry. And next, coming to the table 3, there is enzymes produced from Aspergillus. So we are showing only the Aspergillus species. So that is Aspergillus niger and Aspergillus avamori and Aspergillus oryzae and Aspergillus fumigatus, etc. These are all the enzymes produced from the respective strains of Aspergillus, and this is the fermentation model. That is, so this is submerged fermentation, that is fed batch and SSF, and the submerged fermentation F means fed batch. If it is only SSF means the batch. And so these are all the substrates. The substrates are used for the fermentation process of wheat bran, rice bran, soybean meal. So these are the examples. And table number 4 is FDA approved CHO derived recombinant proteins, and these are all the products derived from CHO derived recombinant, that is animal cell lines. So animal cells are used for the cultivation by using a bioreactor, by maintaining the 5% CO2 and the temperature is 37 degrees C, to produce all the components like tissue plasminogen activator, erythropoietin, deoxyribonuclease 1, beta glucose ribositase, and interferon beta, and all are very important, pharmaceutically important. All the companies, Genentech and Amgen, so Abbott Pharmaceuticals, all are very famous companies, and these are the trade names of these products. So all the products are derived from animal cell culture using recombinant proteins. Next, coming to the important characteristics of industrial microorganisms, the important characteristics of industrial microbes. So the microorganism, we are going to use it. So we had the overview of all the animal cell, plant cell, and everything. And now we are going to focus only the microorganisms, because the microorganisms are commonly used in the fermentation process when compared to animal cells and plant cells. So because It is very easy to grow and very economical, and the media cost is very less, and the fermentation economics is less, and the multiplication rate is very fast when compared to animals and plants. So considering all these aspects, okay, we can give more importance to microorganisms. That is why. So this topic is important characteristics of industrial microorganisms. Usually, the microorganism used for the industrial production must meet a certain requirement. For example, we cannot take all the organisms for the commercial productions. Not like that. Okay, we have to consider each and every point. And I gave the different segments. Okay, first point is the organism must be able to grow in a specific medium and should preferably not require the growth factors. For example, if you are preparing the medium, that medium should be very economical cost and should have basic the components of the media, that is the carbon source and nitrogen source, not much complicated products required for the preparation of media like preformed vitamins, nucleotides, and the acids and all. So all these will increase the cost of the production, and it is not recommended to use the much cost. That is increased cost. Okay, that is if you are using very costly ingredients, that will shoot up the cost of the product also. So additional growth factors may increase the cost of the fermentation, and hence that is the that of the finished product. The first point is simple medium, that is preferable. Second one is the organism should be able to grow vigorously and rapidly in the medium of use. So we have to use the organism, okay, that should grow very vigorously and rapidly. That is, the multiplication rate should be very high. You should not choose the microorganism usually that grow very slow. If it is very slow and you have to wait for so many days, and each and every day you are putting a lot of money for the production, and including the manpower, electricity, so many requirements are there at the time of production. A slow growing organism, okay, that is regardless of its efficiency, in terms of the production of the target material, okay, and could be a liability. Okay, this one, which one, the if it is very slow in growth, this will decrease the efficiency of the production. In the first place, the slow rate of growth exposed to the higher risk of contamination. If the organism is growing very slowly, automatically, if any small contamination, for example, very maybe one or two microorganisms may be contaminated by inserting the inoculum. So that will dominate your organism, because your organism is growing very slowly, and the contaminant one that will grow and spoil all the fermentation process. So your organism should be the fast enough or grow very rapidly when compared to any other organism. If it is not possible, no problem. But if you have an option, you can choose. Okay. Second one is the rate of the turnover of the production of the desired material is lower. So in a slower growing organism, the rate of the turnover of the production. That means if the microorganism growth or multiplication rate is very less, and maybe this is so this will reduce. Okay, the desired material is lower in a slower growing organism. That means the capital, that is investment capital, the person are involved in the production. So all the matters, including the manpower, electricity, and the cost of the media and everything will be extended. So considerably lower profit will be happening. Okay, this is there is a separate economics part. There is a fermentation economics, so that is not mostly viable. So we have to choose. The second point is we have to choose the organism must grow very vigorously or very rapidly. Third point is not only should the organism grow rapidly, but it should also produce the desired material. So you should not say that that is the organism is very fast, at the same time my product is formed is very less, or otherwise very slowly it is produced. So we are cultivating the organism to get the desired product. So if the organism is growing faster, at the same time you should get the more metabolic products or your product of interest. So that is also should be parallel. So the third point is not only should be the organism grow rapidly, but it should also produce the desired materials. And fourth point its end product should not include the toxic and other undesirable materials. The end products, maybe at the time of the finishing the fermentation process by batch or fed batch or whatever may be, and if it is producing the toxic metabolite or otherwise undesirable materials, if it is interfering with the downstream processing, the protein purification, or some contaminant may not be removed from the media. If it is like that, okay, maybe a big headache for the downstream processing process. We can produce by upstream, but the downstream is a very difficult task. So the end product should be free from any toxic or undesirable materials. Okay. And the fifth one is the organism should have a reasonable genetic and physiological stability. So this is very important because if you are working with a microorganism, the microorganism is the strain should be maintained. The mother culture has to be maintained. From the mother culture, you have to make a subculture, and each and every batch, okay, you have to do it. For example, if a person is taking the subculture and from that, okay, he is taking another subculture, the further subculturing of the microorganism, the original, the genomic content may be modified. Automatically, your product may not be formed sometimes. For example, if your product is genome based, there is no problem. The possibility of getting the further subculture, okay, will not modify much of your the product flow or something. If it is plasmid borne or plasmid oriented, the subcultural process, okay, may decrease the plasmid the turnover numbers and the copy of the plasmid. Automatically, okay, this will reduce the product yield, and sometimes, okay, this plasmid may be disappeared from your organism if you are doing subculturing continuously. So this may be a problem. Okay, we have to ensure that, okay, this organism should have its own genetic material, and the genetic material should not be modified or otherwise should not be disappeared, okay, if it is a plasmid. So the maintaining the mother culture is very important, and the organ should have the reasonable genetic and physiological stability. This physiological stability is very important. So the result could be reduced yield of expected material, that is the production of an entirely different product or even a toxic material can be possible. Okay, and the sixth point is the organism should lend itself to a suitable method of product harvest at the end of the fermentation. The organ should lend itself to a suitable method of product harvest. For example, an yeast and a bacterium were equally suitable for manufacturing a certain product. It would be better to use a yeast if the most appropriate recovery method was centrifugation, because the yeast can be easily separated by centrifugation method. So this is due to the bacterial diameter is approximately 1 micrometer, while yeast are approximately 5 micrometer. So the faster sedimentation rate will facilitate the faster recovery and faster purification or a faster downstream process. If you have two sources, one source is good for downstream processing means, but equal, both are equal means you can choose that one. So this is also better. And the next point is wherever possible, the organism which have the physiological requirements which protect them against competition from the contaminants, okay, should be used. An organism with optimum productivity at high temperature, low pH value, which can elaborate agents inhibitory to competitors, has a decided advantage over others. That's a thermophilic efficient producer would be preferable to a mesophilic. For example, the first one is the physiological requirements. Physiological requirements, temperature, pH and all. An organism with optimum productivity at high temperature, maybe if you are producing it at normal room temperature or otherwise extreme high temperatures or very low pH value, that is more stable when compared to all the other components. So if it is there, okay, maybe you can use the thermophilic efficient producer, because that is more thermostable when compared to thermolabile organism. Okay, and if you have two options, choose the thermophilic efficient producers would be preferred to a mesophilic one. Okay, and the next point is the organism should be reasonably resistant to the predators such as Vibrio species and bacteriophages, And okay, all these are very important. That is, the organ should be reasonably resistant. That is, if it is not resistant, if it is prone to bacteriophage, maybe the infection, or otherwise the further the rupturing of the cells may happen, means there is no use of producing the organism, okay, by creating the biomass and getting the products out. If any small contamination, for example, if it is a bacteriophage, okay, it's also grown, and automatically it will lyse all the cells, okay. And your organism should be the phage resistant yet high yielding strains of the organism. So that is a very important point, okay, we have to keep it in mind. And the next point is, where practicable, the organism should not demand large amounts of large amounts of oxygen as aeration. So the aeration is a big problem, okay, in the, because we are going to study, okay, the separate chapter as oxygen mass transfer. So that is maybe after the fermentation kinetics, okay, we'll study, okay, that one in bioprocess technology. And here there is a point we have to consider, that is, the organism should not demand large amount of oxygen, okay, as aeration. For example, if your organism is more aerobic, that means, okay, it requires more amount of oxygen when compared to all the other organisms. So your entire power demand goes for vegetation and aeration, spargeling, everything, because they need more manpower and more power consumed. Approximately the 20% cost of your finished product, okay, goes to that, okay, the pumping, compressor, and also the machineries for vegetation. So the mass flow controller is a very important component of the fermentation process, and that will struggle a lot to maintain, okay, this the dissolved oxygen level, okay, within the bioreactor vessel or fermenter vessel. The maintenance of dissolved oxygen, availability of the oxygen to the organism is called as a dissolved oxygen level. And maybe if your organism needs a more amount of dissolved oxygen, maybe more than 80%, it is very difficult to maintain 80%, and you have to pump a lot, okay, and storing, okay, all the that is pumped air, okay, into a compressor, and compressor will pass the air into the sparger, and after that, okay, this the complete the dissipation of all the bubbles, okay, into the fermenter vessel by agitation using proper impellers. So many complications are there. So better we can use a microorganism utilizes the normal oxygen concentration, that is the dissolved oxygen. Lastly, the organism should be easily amenable, okay, to genetic manipulation. For example, if you have an organism, so if you are identifying or enumerating the organism for the production of your the product of interest, okay, that should be more amenable, that is more possible for genetic manipulation. For example, if you want to increase the copy number of the plasmid, if it is a plasmid borne, maybe the enzyme or some other secondary metabolite may be related to the plasmid, you can increase the copy number of the plasmid, okay, of the particular organism, or otherwise maybe you can modify, okay, the genetic composition, that is the genetic manipulation by recombinant DNA technology, that should be possible for your organism. So choose the organism, okay, based on that. If you want to go for the further steps, it may be very easy for that. Okay. If you are getting an organism, that organism, okay, maybe okay, production is okay, but at the same time, okay, increasing the production. So increasing high copy number, or otherwise increasing the copy number of the plasmid, that may be a problem, means it's very difficult. You may be stuck up with some of the aspect, okay, with a limited product yield. So that should not be there. So try to identify if it is not possible, no problem. And if it is possible, maybe you can choose the organism, okay, based on that. So these are the 10 aspects. Okay, all the aspects are very important. Okay, all right. So next topic is fermentation kinetics. I'll give the introduction in this lecture, and maybe the lecture number 6 we can go in deep into this. So this fermentation kinetics means, I hope everybody studied about enzyme kinetics. So this is the different version of enzyme kinetics. The both kinetics are same, but there, okay, we used enzyme, and here we are going to use fermentation. That's all. and maybe you can refresh about what is enzyme kinetics. So what is the reaction of the enzyme, and what is the value of Emax, and what is the value of Km, how to arrive the value, and so what is the equation, and what is the Michaelis-Menten equation. So you can recollect all the equations of the kinetics part. So before coming to the lecture, so now I'm going to stop this